Hi, welcome to Neuroverse,
a podcast hosted by Clara
and Carolina, where we discuss all matters from neuroscience to philosophy and beyond.
Today's episode is on science misinformation, the third part of our series. This episode will be specifically on real science, desperate scientists, inspired by a current topic right now in the specifically neuroscience field. So let me begin by providing some background of what inspired this episode. Recently, a story came out about some science misconduct that has happened in the neuroscience community, and we wanted to address what exactly happened as well as why and what is being done to prevent this in the neuroscience community. So it had to do with some researchers studying Alzheimer's disease. So recently an investigation was launched by a neuroscientist at Vanderbilt University, Matthew Schrag, as he studies the mechanisms underlying Alzheimer's disease, but he was very skeptical about some findings that had come out in 2006 and were very high impact and inspired the development of a drug which is now being promoted for Alzheimer's. The drug is simufilam. It's created by Cassava Sciences, and what it does is it repairs a protein that is supposed to block these deposits of a pathological protein called amyloid beta. And amyloid beta has been one of the best leads for Alzheimer's research, in that the amyloid beta hypothesis was conceived by John Hardy and colleagues back in around 2000, very long time ago. And so much research has basically used animal models containing this pathological protein to try to understand what's happening and how it can cause the disease.
Yeah.
So most research has suggested that amyloid beta misfolds and it then starts to aggregate into these clumps and what we call plaques in the brain, and this is what prevents neural transmission and slows down neural processing and causes these cognitive deficits. Well, there was another team at the University of Minnesota that their whole research basis was that amyloid beta in a different form, which is actually soluble, but they are what we call oligomers, so it's also another misfolded form. These soluble amyloid beta oligomers accumulate extracellularly, so like outside of the cells, and they cause memory deficits. And this is another new leading hypothesis, because so much work has been done in amyloid beta, no drugs have come out of it that have been successful, and now people are like, oh, maybe it's the silent soluble oligomers that we never thought of. Yeah. So this is becoming more popular as a theory, and the simufilam drug is based on this research. So Matthew Schrag started investigating the work of the group at University of Minnesota who have been studying this for a while now, and he began to suspect that there was misconduct in their research, in that they were publishing these images that didn't look that believable, and it looked like they had interfered with the data. Not that the images were completely false, but that they changed them and altered them by themselves to exaggerate the results. And so overall, there was then a whole investigation by Science magazine which provided strong support for these suspicions. Now, it's important to say that the oligomer that this lab was focusing on is called A beta star 56.
Yeah, a really specific strain of the oligomer.
Yeah. It's important to note that many other scientists have actually tried to replicate their findings that this A beta star 56 causes memory deficits, but they have been unable to. Yeah. And this includes Dennis Salko, who was one of the founders of the amyloid beta hypothesis, who has written many papers about the hypothesis in general. And so overall, this caused a huge uproar in the neuroscience community, and specifically, of course, the Alzheimer's community, because a lot of studies have been carried out based on these findings, which were published in Nature, arguably the most high-impact journal in 2006, which was quite a long time ago.
Yeah.
And so reputable neuroscientists have commented that if anything has come out of this whole situation, it's that resources and funding has been wasted.
Yeah.
Because if these results aren't true, then they have fueled so much extra research for nothing.
Yeah, and not only that, a lot of other studies, as you've mentioned, that built their research on the backbone of these findings that also are probably now very much inconclusive because...
Exactly.
And then again, wasted funding.
Yeah. It's also brought a lot of attention to the problem of reproducibility in neuroscience research, as well as the problems in neuroscience publishing and journals and the pressure that scientists feel.
Yeah, that's a really good point. I think that the importance of reproducibility of science is something that a lot of scientists are aware of, but in reality and in practice, it's very difficult to achieve.
Yeah.
But there are really good efforts to do that.
Yeah.
I think that, for example, with the International Brain Laboratory is this new foundation that their main and only goal is to have reproducible electrophysiology data. So electrophysiology is a way to measure neuronal activity, but specifically looking at the current and voltage. And so the tool Clara and I are mainly used to using is patch clamping, which is where you record activity from one single cell. And due to the strenuous difficulty of the nature of patching, N values tend to be quite low. And then on the opposite spectrum of still electrophysiology tools are things called neuropixels, which are a very thin fiber that you can put in the brain and it can record activity from millions of neurons. And now the problem is the opposite, is that there's a lot of data and we need to really think about how reliable the data is, how to filter out the noise, etc., etc.
Yeah, and what it means.
Exactly. And because of these variations, a lot of scientists have found that electrophysiology data is difficult to be reproducible, and so this International Brain Laboratory has been created where they have a very open science approach. People put a lot of their resources and findings and preliminary data and GitHub type links and things like that to allow this reproducibility to occur. And I think this should be extended to all other branches of neuroscience, not only electrophysiology, but also clearly molecularly there's still challenges with reproducibility.
Yeah. This is specifically, so the problem with reproducibility, as you said, is throughout all science, but it's specifically a problem with neuroscience because not only do we always try to limit the number of animals we use if we're doing animal research, but within an animal, you then record cells or neurons or brain regions. And so there's this problem called pseudoreplication, where you're taking multiple values from the same organism. And you can treat this data as individual data points, but technically it's still from the same organism. So you have to take that into account in your analysis. And that involves doing multi-level analysis, which is quite complex. And plus having more N numbers, but then you're limited because you're trying to reduce the number of animals you use. It's quite a big of an issue. So you need to really find the balance between your N numbers and your optimal data analysis tool to come to the right conclusion. But linked to what you also said about this International Brain Laboratory, which I had not heard about.
Yeah.
There's a really good article about the A beta star 56 story in The Atlantic. And they also mentioned, just as you said, that what would prevent this from further happening in the future is to have a culture of transparency, where honest errors are readily corrected. And something they highlighted, which is so true, is that at the moment, there's no incentive for scientists to come out and speak against other scientists. And they also talked a bit about science communities can be very small, which is true. Not the whole neuroscience community is big, but then within that, there's so many subsections and those communities are small. And
Definitely.
so it's rare that a scientist, it takes a lot of courage.
Yeah.
And firm beliefs to come out and say look, I think something wrong has gone wrong.
Yeah.
And nobody wants to be shaming other scientists. And also this
a big name.
Yeah. And also the danger is that this has brought to light the problems within scientific research, which is already something that the public is always criticizing scientists
and skeptical about
and
already.
skeptical about. And so it's like even more important to use this as an opportunity to further look back and say, okay, what are the tools that we have to improve reproducibility and to improve the reliability of our experimental methods and analyses.
Yeah. I think related to what you were saying about, there should be a better system of keeping research already out there into check and verifying if it's correct. And Schraug initially discovered all of these and errors and altered data in this site called PubPeer, which is a site where other scientists do comment on these types of things. But again, I think this process is quite informal in the same way that like peer reviews also has their own issues. And I think there's a bit of a systemic issue in the way that things are published, and the fact that, you know, scientists are often not rewarded about their peer reviewing jobs, and it just adds a lot of hours and workload onto scientists. And another thing related to reproducibility is a lot of people avoid publishing negative results because they aren't necessarily groundbreaking, but they are incredibly important. And a lot of people who found that the amyloid beta star 56 didn't publish those results because, one, they didn't want to go against a big name, and two, they didn't want to publish negative results because that's not interesting.
It's been proven that the scientific journals have a large bias towards publishing positive results. But that also speaks to so besides the problem of the misconduct of this scientist, a bigger problem really is the pressure of how scientific journals work. And as the same article in The Atlantic put it, there's a publish or perish mantra that guides scientific publishing. And so there's just this huge pressure on scientists to always find positive results. Like you said, if you don't find positive results, you're not even going to submit it because it's going to be rejected. And if you do, to get into a high-impact journal such as Nature, it's seemingly more to do with your results than anything else, than your experimental method or analysis pipeline or whatever. If you have something really, really groundbreaking and different, then that will make your career. And that's just how things work.
And related to that, not only the publish and perish mantra, it relates to the fact that, if you get into a high impact journal, then your lab is reputable, and then you can apply for grants, funding. You get more funding, and you can stay alive. You know, if you don't publish, you won't get any funding, and that will be it. That's the end of your lab. And so there's an enormous pressure with a lack of funding, a pressure to publish. Science is not getting rewarded for all of the extra work that they do, such as peer review. As much as science misconduct needs to be stopped, we do need to look at the systemic pressures that sometimes do drive this scientific misconduct.
And therefore it's not surprising that this particular story occurred within the Alzheimer's field, because there is so much pressure after over 30 years maybe of this amyloid beta hypothesis for something to come out of it. Like, there is not yet a single drug that is effective that targets, well, anything in Alzheimer's, but specifically amyloid beta either. And so if you can have some sort of hope, then, you know, you want that hope.
This relates to a couple of quotes that were in the science article that published this investigation about how you can cheat to get a paper, you can cheat to get a degree, you can cheat to get a grant, but biology doesn't care. Like, data is data.
I think that was a quote by Matthew Schrage.
But I think that's the crucial bit. Data is data, and I think scientists do get desperate and try to like see things when there aren't any. And an example where I saw this occur was actually in FENS, which is the major European neuroscience conference event that happened in July in 2022, where Natalie Rochford presented their current research that she's working on, where basically she discovered that with food scarcity, predictive accuracy decreases. And this has major implications for so many behavioral studies that use food scarcity to drive reward and or fear and motivation. And there was a scientist that came up and asked, well, what do you suggest, in a very confrontative way? And Natalie, of course, was like, well, that depends on the study, that's very conditional to the type of question you're answering. But essentially the message was to take into account the fact that food scarcity does alter the data. It has different effect on male and female mice, their physiology. And if you can use different methods, or at least be aware of the limitations, that's really important. But a few scientists did seem to be quite confrontational about this new realization of what food scarcity has on behavioral experiments. But like Matthew Schorg said, data is data. And now we as scientists need to take that on board and adapt that and make sure we design better experiments that take all of the information we have available into account. We can't turn a blind eye on what the data is telling us.
Yeah, so scientists obviously have a huge responsibility as scientists, because scientists are responsible for the future of the world.
Despite what the funding suggests.
Yes.
And appreciation.
Yes. And efforts are definitely being put into constantly improving the way that science is done, and also how the science community functions, despite the difficulties that the funding presents us with.
Kind of also, just to reiterate a little bit, in that even though amyloid beta star 56 has been flagged as having the research around it having misconduct, we should be very careful to not, again, over exaggerate, as I've seen a few quote unquote clickbait articles do, basically saying that all amyloid beta research is now spoiled. And that's,
it very much is not.
It's so not true. And just to give an example, the discovery that oligomers could potentially be a cause for Alzheimer's is still, the research is very solid in that there's a very high percentage of oligomers being the main protein misfolding type that creates holes in the membrane, and this is particularly seen in ion channels and affecting NMDA and AMPA receptors and things like that. So the research around it is solid, but we're still figuring out how everything works. But clearly, this star 56 amyloid beta oligomer type does have some misconduct.
It's actually unfortunate in many ways, because they intervened with the data in order to exaggerate an effect. But who's to say what the effect was if they didn't? Mm-hmm. Because I'm not sure if there would be no effect at all, or if it would be smaller,
Yeah.
And nobody really knows unless it's reanalyzed, perhaps.
Yeah.
Yeah.
And I think, Simon Schultz tweeted about the fact that he believes that there is a lot of hype now around the fact that a lot of oligomer research is discarded, and he was a bit confused as to why so many people were making a big deal out of it, because in his eyes it's a small fraction of the oligomer research out there. So just to reiterate that not all research around Alzheimer's has been tainted.
Yeah. On that note, we'll wrap up today's episode on Science Misinformation Part 3, Real Science, Desperate Scientists, which I just love to say. We hope you enjoyed this episode.